So for this last couple of weeks, we're gonna basically be finishing up the rest of the material. We have meiosis and genetics is this week, and then we'll be getting into the last unit, which is about energy. So it's energy and enzymes, and then photosynthesis and cell respiration, which is a lot for the last week, but hopefully you've learned about those things before, so it hopefully will be a lot of review. So today is our day for meiosis. I think we can get through that whole lecture. The ICA is here and we'll have a chance to work through a lot of that today in class. And then for the lab part, I came in on Saturday morning and I moved everybody's adults 2 new plates. So they're on their second day plates. What you're going to be doing today, and I haven't even looked to see what stage they're at, but hopefully we have L4s that are the progeny of the ones that you put on. So by this time, we did that Thursday, so Friday, Saturday, Sunday, Monday, Tuesday, we definitely should at least have L4s, or they might be slightly older. Since we only have one chance to do this experiment, we're going to take whatever it is that we have. If they're L4s, that'd be ideal. If not, we'll pick the youngest adults that we can find. And you're going to have to do 5 per plate. So you have GMC 101 and you have the N2. So that's 10 worms. So that's not too bad. I will do the SMB1 negative control, and the PFN1 is the positive control. So I will deal with your controls, and then you just have to deal with your strains. So that we'll do. And then the other thing I want to, because our poster's coming up next week, I want to give time this week to work on that. So for today, I thought what we could think about is what we want to use for results. So we got to figure out basically what figures will tell the story. You have a lot of data so far, right? We've done, you ran gels, you sequenced. So all these different pieces can be included. It doesn't just have to be the paralysis assay, but I would say the paralysis assay is probably a pretty important part because that's really what the research question was about. So that's for today. The rest of the week we're doing the scoring of the paralysis assay, day 1 and day 2. And then what I actually wanted to talk to you all about is for Wednesday, I have on the calendar to do a genetics discussion. And so I have one that I've often done, but if you've taken me, like Ken has been in my class, in my 160 class, we did this genetics discussion. So I was going to give the class a chance to vote on the topic. It doesn't have to be that one. So what we did in 160 is this pre-implantation genetic diagnosis, which is a technique where they take an embryo, so they do IVF, they fertilize an egg outside of the body, and then when it gets to a certain stage, they take a single cell and they can do testing on it. They can look for diseases, like if there's a disease that runs in your family, you can test for that. And then you use that to select which embryo to implant. So it's a way to select, it's not so much traits, but it could be disease, but you also could select traits using that technique. So that discussion is kind of looking at different scenarios and deciding when we should do that, if at all, should that be regulated, that's kind of the genetics discussion. So that's one. The other ones I was going to show you, there's a whole bunch that are here that are really interesting topics. Let me go to the ones I was thinking of. So this one I think is an interesting question. It's about, and you've probably heard about this. So with athletic events, the testing for sex. So, you know, if you have someone who's competing in a woman competition, right? And how do they determine that this person can compete in that competition, and will that be fair? So this takes a look at it. It's more of a comprehensive look at all of the different parts of sex from sort of a biological standpoint. So that's one question that we could look at. The other one is about privacy with genetics. So as more information, we talked about like 23andMe, if you're going out and getting these tests done, what kind of concerns do we have about privacy? Because obviously those can reveal some risk that you might have to get a disease or something, and that could affect whether, you know, if it gets out there, whether or not health insurance companies want to insure you. Because what if you have a very good chance of getting something, you know? So this is about privacy and genetic testing. So there's the PGD, sex and athletics, and this one, privacy and genetics. And I can think of a fourth one that I've looked at, which is genetics of mass killers. So there was one time I did this in my genetics class, looking at some of the mass shooters, and they kind of take a look at, are there genes that predispose somebody to be violent, and so we could do that topic. So there's basically 4 topics. So if you want to think about it for, and how can we vote? Maybe I'll write up here. I'll write them up, and then maybe I'll just have it be anonymous. You can, on a piece of paper, I can hand out. So PGD is the genetic testing of the embryos and deciding which ones to implant. Sex and athletics is the complicated decisions about how do we, what's fair and how do we test for sex, for competition. And third is privacy and genetics, genetic testing. And then fourth would be genetics of, I would say mass killers, or that's not a good word. You know what I mean.
Murderers.
Yeah.
Street killers.
Okay, so those are the 4 options. Let me see if I have like sticky notes or something. Or maybe, do you all have like a little scrap of paper? Okay, you can close your eyes and go anonymously. Whatever you want to do. Let me go get sticky notes and then I'll just hand them out.
Hands up so hard for live. I should be done.
It's just like the discussion.
So you can write on the inside of the card and then hold it in half of it. And hopefully it's not even high.
I have a question. What if I have like 2 in mind that I want to choose? but
So
I
you
don't
could
know
put-
which one.
Oh, you don't know which one? Maybe,
Yeah.
so you don't have a preference?
I mean, like, I have a preference, but I have 2 in mind.
Okay, you can put them both.
Okay.
That way, if we need to break a tie, we can look at. Yeah, if you have top 2, you can put top 2, and then maybe that'll. I don't mean to make this complicated, but. Okay, so that'll be our Wednesday discussion, which for genetics discussions, I usually give you something to read ahead of time, but it's fun reading because it's not like all sciencey, it's more like applied science. And then in the discussion, usually we do small groups, and I either give you a scenario or something, you have a small group discussion, then we have a large group discussion. All right, so I'll tally those up and then we'll let you know what we're gonna do for our discussion on Wednesday. And then Thursday, hopefully we're done with genetics and we're starting on energy and metabolism, and I'm definitely gonna give you a lot of time to work on the poster on that day to put final touches together, because the next week is when we actually have the poster session. Anybody have questions about the week plan? Okay. So, then we're gonna get started with meiosis. Oops, not the one. Oh, and I think I told, let me try to start a Zoom call. I'm gonna record this as well. Because I know Brianna is out and she wanted me to record. Somehow I'll try to do this off of Zoom. Oh, yeah, so I'm kinda like, and then I guess I'll minimize it. I've not tried this before. Okay, we're gonna share screen and record. Where's the record?
Go to more.
Oh, thank you. Record the live, okay.
So it was a simple, I did
Okay,
co-op like study, so we have like 4 films that you could choose. I know.
PowerPoint, all right.
Voting there, but.
Alright, so this is chapter, I think this is chapter 13 for this material. So it's kind of an extension because we've already talked about mitosis as one form of cell division. Now we're going to talk about another form of cell division that is used specifically to generate the gametes. So the job of meiosis is to, well, it has to do a couple of things. One is that it has to split the total genetic information in half because you have to go from a diploid cell to a haploid cell, which can then recombine to form a diploid cell. And the other thing that meiosis is trying to do is to generate genetic diversity for, to have variation. So this is used for sexual reproduction. So we're going to compare and contrast sexual and asexual reproduction. And then again, we'll go over some chromosome terminology. I think I introduced the idea of chromatids and ploidy, but we're going to talk about some of those definitions. And then we just go through the steps of meiosis and compare that to mitosis. Okay, so those are kind of the main topics for today. So for the first comparison here, sexual and asexual reproduction. For each of these, they vary in these different categories. So the genetic identity of the progeny that are created, the phenotypic variation of the progeny, and whether or not each form of reproduction would lead to survival. So for the first one, genetic identity, if you have asexual reproduction, what do the progeny look like genetically? Are they similar to or different from the parents that create them? Yeah, in fact, identical, right? So they are genetically identical, and that's because an exact copy of the genetic information is made. For sexual reproduction, how about the progeny in this case? Are they identical or different from? Yeah, they're gonna be not identical, they will be variable from the parents. And then if you look at the whole, basically the litter, the progeny set from asexual reproduced individuals, they're gonna be, have no phenotypic variation because they're all clones. You can think of many, a bunch of clones of the parents. So there's no phenotypic variation. Whereas, as you know from your own families, siblings in a family are different. So there's high variability of phenotypes, of the traits of those progeny. Now, which one of these do you think could survive in changed environmental conditions? Which one has a better chance to survive if the environmental conditions change? Yeah, so why do you say that? That is right. It is sexually reproducing individuals have higher survival. But why, how can we explain that? Yeah, Daphne?
Animals, no, for those who were sexually reproduced, they have more genetic variability, which means a higher chance that one mutation is beneficial in the changed environmental condition.
Yeah, exactly. There's got to be some individual in that population that has traits that are suited to that change. So that's exactly right. So for asexually reproducing, we say they have low survivability and low adaptability because adaptability comes from the variation and having a whole bunch of individuals with different traits in your population. So then likewise for sexually reproducing, we would say high survivability and high adaptability. For the definitions, I would say you could, asexual reproduction is any form of reproduction where the offspring inherit DNA from one parent. So for asexual, we'll just write progeny inherit DNA or genetic information from one single parent. And then for sexual reproduction, it's any form of reproduction in which they inherit genetic information from two parents. I can't think of any examples where you get it from more than two, but maybe there's an example out there where that's the case. But progeny inherit DNA from two parents. And for, a lot of organisms, I mean, it's either asexual or sexual reproduction, but there are cases where you can have both. So some organisms have examples even with plants. You can have clonal offshoots that create clones, but at the same time you could have sexual reproduction in plants in the same species. The bee I put a picture of there because bees are interesting. Females, they get all they collect all the sperm they need for a lifetime, and then when they fertilize their eggs, those become females, the future females of the colony, and then the ones that are not fertilized are males. So the males in bees are actually haploid, they are not diploid. So there's a lot, if you look across the animal kingdom, you see a lot of different variations on this. Okay, so this does introduce kind of a paradox though. Oh, you can't see the top. Let me move this thing, the bar. The paradox of sex is that let's just assume that we have two populations, they live in the same environment, so they live in the same niche, and they're competing for resources. And let's just imagine that both of them, each individual that is capable of producing progeny can produce 4 progeny. So for asexually reproducing, that first single individual can produce in generation 2, 4 individuals, and then each of these can produce 4. So by the 3rd generation, you're up to 16 individuals. If you look at a sexually reproducing population that lives in that same niche, the female is the only one that can bring forth progeny, right? But has to have a male in order to do so. So let's say that this individual has 4. The likelihood, just by chance, 50% should be female and 50% should be male. So in the 2nd generation, there's typically only 2 individuals that can still bring forth progeny, that can reproduce. So we go from 1 to 2, and then in this next generation, we have 4 individuals that can actually have progeny. So which one of these seems to be more efficient at producing individuals? Yeah, you can see it's prolific growth of that population, right? And so to have a sexually reproducing population is going to be less efficient. So the asexual reproduction is faster, more efficient than sexual reproduction. But what would be the benefit then? Why would you go to the trouble? I'm just going to move this thing out of the way. Why can't I get my, oh, there. What would be the benefit then of actually, why do we have to have males? If, it seems like they have a higher cost. What is meant by the phrase cost of males? We have males in the population, but they're not capable of bringing forth additional individuals. What's the benefit of having males?
Good question.
Good question. Males, you wanted the question. What do you bring to the, what is the benefit though? There is a benefit of having males around.
Genetic diversity?
At least one.
Are you sure?
What'd you say?
Genetic diversity?
Yeah, so that's a way to introduce genetic diversity. So even though it seems like this is costly, because half of the population can't actually, bring forth progeny, but the benefit is that they're going to bring all of this genetic diversity in. So if let's just look at this question, if they were competing and everything's the same in the environment at this point in time, which one would be more successful, do you think? They competed for the same resources in an unchanged environment. Yeah, that would be this one, because they're going to outcompete. They're going to have more individuals to get more of the resources. But if there's any change in the environment, then which one has the advantage is going to be this one. they've actually looked at this with different, there's species of worms, like the nematodes. There's some that are sexually reproducing, and when they compete for the exact same resources, if the environment is unchanging at that time, the one that does best is the one that can self, that can, doesn't need to mate. But then if you introduce a change, then the other species has the advantage and sort of takes over that niche. So there's a couple of theories on how this evolved, why sexual reproduction evolved. The first idea is that if you have sexually reproducing individuals, you have diploids that have two sets of chromosomes. That's what diploid, di is two, and ploid, this word means set of chromosomes. If you have a diploid, there are two copies, so you really have a backup in case you receive some kind of an allele that is deleterious or harmful to the species. You have a backup copy to provide you some protection. So this idea is called the purifying selection hypothesis. That's one of the reasons why this could have evolved. And then the other one is about what we just talked about, the changing environment, that if there are changes, which as we know there's constantly changes in the environment, this provides the diversity that you need to have protection. Do you think it's one or the other, or do you think both of these could coexist as being sort of explanations for how this evolved? It's probably not one or the other, right? They could both explain how this evolved. All right, so any questions so far? Just the basics? Okay. So, coming to the terminology here, and this, I think people think they understand ploidy, but then a lot of times when I give you these charts that ask you to think about the ploidy numbers, that's when you realize like, oh, maybe I don't know if I understand what ploidy is. So ploidy, the word itself means one complete set. A mono, or haploid, not monoploid. A haploid means that you have one set of chromosomes. The number of chromosomes in that set can vary. So we use the letter N to represent the total number of chromosomes in one set. For humans, does anyone know what the N value is? For one set. So there's, or if you know the total number of chromosomes, go ahead and work backwards. 46 total, right? So that's for two sets, so then it's 23 chromosomes in a set. If we look at Drosophila have 5 chromosomes in their set. So the point I want to make is that this number can vary, but what the N represents is that you have this number of unique chromosomes in that set. And they have different sizes, they have different genes on them, so that's what makes them all individual chromosomes. The other terms here, we have polyploidy, which means multiple sets. Usually we use the word polyploid if it's more than 2. For 2, we say diploid. So haploid for 1, diploid for 2. More than 2, you would say polyploid. Does anybody know an organism that can be polyploid? Or just generally which organisms tend to be able to tolerate this? It's usually, what did you say? Strawberries, yeah, that's one. So plants, actually, there's a lot of species of plants that have duplicated genomes and they can tolerate that. You've never, ever seen a triploid human being. That's not, if that were to happen, let's say that an egg got fertilized by 2 sperm, polyspermy. That can happen accidentally, but the body will spontaneously abort that. So that's called a triploid, because it would have 2 sets from the sperm and 1 set from the female, from the egg, but that will be spontaneously aborted. And so humans and most other animals don't tolerate polyploidy, but you will see polyploidy if you look at different plant species. Okay, and then the other term that's important that we'll talk a lot about today is aneuploidy, which just means irregular number of chromosomes. Now this does happen in humans. Can anybody think of a condition where you have the wrong number of chromosomes?
Down syndrome.
Yeah, Down syndrome. So that would be trisomy, so you have an extra copy of your 21st chromosome, so that's one of the few that does produce a viable offspring. There's a couple of others that you can see, but they're usually lethal, like trisomy 13 and trisomy 18. If that ever happens, then usually the baby will die shortly after birth, or it will be a stillbirth. So trisomy 21 is the one that is tolerated and produces a perfectly viable offspring. Okay, so this is a nice little video. I don't think I'm going to go through it right now, but if you feel confused about ploidy later on, like when we do the worksheet, I would come back to this one and watch. It kind of explains it again in very simplified terms. All right, so concept check here. So for humans, we are diploid. We just said N is equal to 23. So how many chromosomes are in the egg and the sperm? 23. And then when they come together, we get 46. So 2N is equal to 46. For Drosophila, I told you that N is equal to 5. And so the egg and the sperm would contain 5, and when they come together, they would have 10. This one is one I also use as an example in Bio 160, but strawberry can be tetraploid, hexaploid, octaploid, decaploid. So this is 4, 6, 8, or 10 copies of the chromosomes, and how many chromosomes are in one set is 7. So they can have up to 70 chromosomes. So this is why, I don't know if any of you ever purified DNA out of a fruit. This is why strawberries is a really good source to use. This and bananas, those are good if you want to actually purify the DNA. It's a good source. The other interesting thing I like to share is that usually if you go get strawberries that are those big giant strawberries from like Fred Meyer, usually those actually they have octaploid or decaploid. And the reason they get so big, they're hybrid varieties. But the more chromosomes actually produce the larger fruit. If you go to the farmer's market, their strawberries tend to be smaller, right? But which one do you think has more kind of essence of strawberry taste to them?
Smaller ones.
Yeah, those smaller ones. So what gets diluted out when you get big strawberries is sort of the flavor that is produced. So the farmer's market ones usually have fewer chromosomes. Okay, so other terminology, we said this yesterday, chromatid, we're going to use that to be a single strand of DNA. And it doesn't necessarily have to have a copy to be called a chromatid. I think the textbook kind of refers to sister chromatids, so they make it sound like you have to have the copy in order to be called a chromatid. But if you just have the single strand, we call that a chromatid. When it gets duplicated, now that's called two sister chromatids. What I like to emphasize here is that this is like a Xerox copy. It's not new information. It's literally like going to if you have a book and you make an exact copy of that book, it doesn't mean you have 2 titles now, you just have 1 title, but 2 copies. Now, homologous chromosomes, these are 2 chromosomes that have the same length, so same size. Shape is shape is a weird thing to think about, actually. I would say the same staining pattern rather than shape. But the same content, which means they contain the same genes along the length. The difference here, do they have to have the same DNA sequences on those 2 strands? No, they can have, so you can have the same gene, gene A and gene A, but you could have different versions of the genes. So would we call are these different chromosomes or are they different chromatids? What do you think? Is the information different? If it's different information, then they're different chromosomes. Okay, so when we're counting up chromosomes here, we would say we have 2 chromosomes, we have 4 chromatids in this picture. Up here, that's 1 chromosome and 2 chromatids. So you ask yourself, is the information different in those 2? You could think of it like, both of these are books, and one's in French and one's in Spanish, or whatever, there's different, but the content is the same, right? The genes are the same. So then, for meiosis, we're gonna see an event where the homologous chromosomes come together to form a bigger structure, and that's called a bivalent. I never knew how to say this, but at conferences they say bivalent, so bivalent, or you can call it a tetrad. So when the homologous chromosomes pair up, that forms the tetrad. And they do this, does anybody know why they have to come together? You remember in meiosis? Crossing over. So this will allow you to get exchange of the DNA so that you can actually form new combinations that have never been seen before. These two strands, the red and the blue, those are called non-sister chromatids. And that's where the exchange will happen. So one thing we'll see, when you have an exchange, the outer strands they're unaffected. Okay, so they're not gonna cross over. Let me just kind of do it here. The crossing over happens between two non-sister chromatids. So you will end up, whenever you have a tetrad, if you have a crossover event, two of them will become recombinant. So these two would be called the recombinant strands. And these two are not crossed over, so they're called parental, the parental type. So we'll see this when we get to genetics, and we talk about, let's say that we had a gene A, and on this strand we had, maybe we should color code it, red, okay. So the red one is big A. If you had down here, big B, and then the blue, let's say it was all small, so little a, little b. So you can see that the parental types would be big A, big B together, little a, little b together. The recombinant types have swapped. Oh, oops, that's supposed to be an A. They will swap, so now we have a combination little a with big B, right? So that's gonna generate a combination that has never existed before when you get the crossover event. All right, so any questions on these terms so far? No. Okay, so then the other thing we were talking about, we said that if we have a gene, like in this case, these are our homologs, we have this A gene can have different versions. So when I say big A, that refers to a certain allele. So that version of the gene is called the allele. So for the definition of an allele, it's just a version of a gene. And that can give you a different trait. So in the one that we're looking at here, this would be the gene that encodes eye color for Drosophila. So then you could have one version that gives you red eyes. You can have a different version that gives you purple eyes or white eyes or whatever. Actually, with Drosophila, it's interesting. They have all these different shades of red. They're all like, I don't even know how you would tell the difference. It's like vermilion and coral and anyway. So all these different variations give you a slightly different trait. If you have on your homologous chromosomes the same allele, it's called homozygous, right? And you can have the same version of the recessive allele or the same version of the dominant allele. If you have one of each, then it's called heterozygous. Okay, and you know that comes from what did you get from your maternal chromosome and what did you get from your paternal chromosome. Okay, when we say they're different alleles, can somebody explain what that means for the gene sequence? If we were to actually look at the gene sequence there, would it be? Yeah, there's slight variations in those sequences. And so I give you here, this is the example for red hair. If you have an individual who has red hair, they have this single change. You can see that in one case it's a G, down here it's a C. That single point mutation is all it takes to get red hair. Now red hair is caused by a recessive allele, so how many copies would you have to have of this version? Two. In order, if you have red hair, you have 2 copies of this. If you have another color of hair, usually cool dark, then you would at least have 1 copy of that top. But alleles, they don't have to be in the protein coding region. So I tried to kind of highlight, you could have differences in the sequence that affect splicing you can have differences in the promoter region to affect whether or not the gene is functioning or not. So it's not just protein coding region where the sequence can be different. Okay, so that's kind of the general terminology that we need for meiosis. So now let's look at the actual process for meiosis. Now, we said that meiosis is necessary if you have a sexually reproducing organism because we have to get from the diploid adult, we have to get back down to the haploid in order to produce the egg and the sperm. So meiosis is going to take place in the germ cells to generate the gametes. Okay, so meiosis takes place for males in the testes, for females in the ovary. And this process of producing an egg or a sperm is called gametogenesis. So that is the general terminology to say producing gametes. For females, it's oogenesis. And then for males, it's spermatogenesis. Okay, so at the end of this, I have a section that talks about the differences between oogenesis and spermatogenesis. And they're a little bit different, especially for humans, it's a little bit different between those two processes. Then those are haploid, so they have N. When they come together at fertilization, they create the diploid zygote. And then from that point, this zygote undergoes mitosis. So that's sort of the typical life cycle, right? So mitosis will give you development up to the diploid adult. So now we're gonna zero in on that meiosis phase. So generally speaking, meiosis there's a lot of parts of it that are similar to mitosis, but the main difference, I would say, is that it's the division that's gonna happen twice. There's two actual divisions, and that's meiosis 1 and meiosis 2. Oh, here we go. Okay, so the first division, let's actually do some math here on what this organism has. So we have 2 chromosomes. So we know that it's diploid. How many chromosomes are in the set? By looking at that picture.
How many chromosomes are in the set?
Yeah. 2. So for that organism, it's 2n is equal to 2, which means that each set is only going to have a single chromosome. So this is the most simplified that we could possibly be. In this picture, how many chromosomes and how many chromatids are
2
present?
chromosomes, 4 chromatids.
Yeah, 2 chromosomes and 4 chromatids. At the end of meiosis 1, you can see that what gets divided is the homologs. So the tetrads come together, they're going to cross over, and then they split. And so at this stage, how many chromosomes do we see in each cell? 1 chromosome, and how many chromatids? Still 2, because the sisters are still connected. So in meiosis 1, this is actually called a reduction division. I'm going to write that up for meiosis 1. It's reduction division because you're going from diploid to haploid, 2n to 1n. That's when the ploidy will change, is meiosis 1. And then, for meiosis 2, this is called an equational division. Equational division, because we start with 1n, and at the end, it's still 1n, right? So this is 1n. This is also 1n. How many, for this picture, we said it's 1 chromosome, 2 chromatids. How about at the very end?
1 chromosome, 1 chromatid.
Yeah, so 1 chromosome, 1 chromatid. So this one is very similar to mitosis, where the only thing that's changing is that the number of chromatids is changing. But this is going to generate 4 daughter cells, and they're going to be variable. We're actually not even showing here the crossover event. So technically, we're going to create 2 recombinants and 2 parentals, but all of them are different. All 4 are different at the end of the day. Okay, so now the details for meiosis. So, yeah, I guess we'll kind of go through the major differences mostly happen in the beginning. So this, the stages are gonna be the same, so the prophase, prometaphase, or we can say prophase, prometaphase is late prophase 1. And then metaphase, anaphase, generally what's happening is similar, but there are some minor differences. So look at this picture, I want you to, with the people next to you, try to think of at least 2 things that are different between prophase and mitosis and this prophase and meiosis 1. Come up with at least 2 things that are different.
So looking at this one.
Yeah. The crossover. The obvious ones.
Yeah, the crossing over, yes. And then,
There's no replication in the beginning of the DNA.
oh,
Is that right?
probably. I don't know.
Okay, so just kind of quiet it down a little bit. So what's at least one difference between what we know about mitosis and then prophase 1 here?
There's no synopsis and mitosis.
Okay, that's one. Okay, let's have one from over here. So there's no synapsis. Synapsis is when the 2 homologous chromosomes come together. We never saw that happening in mitosis. Can somebody from over here think of another difference? It's kind of related to synapsis, but what happens after they synapse?
Crossing over.
Crossover? Yeah, so crossing over never happens in mitosis. So here you can actually see the structures where they are crossing over. Now, every single homologous chromosome has at least 1 crossover. You can sometimes have more than 1. So in the picture there, you see 3 of them. I actually think that doesn't happen as often, because usually there's some interference once you've already had a crossover. But at least one time, there's a crossover between every homologous chromosome pair. And so here you can see when they line up, now you can see the evidence of where those crossover events happen. Okay, so prophase are differences that you should mark down in your notes. 1, homologous chromosomes have to find each other and synapse. And 2, they then cross over. So what does the synapsis look like? Well, it's a little bit complicated, where these 2 actually have to find each other. So there's actually been a lot of work done to figure out how do you have homologous chromosomes that locate the other homologous pair, right, to form this structure. When they pair up, they form the tetrad, or the bivalent. So let's think about this question here. If you have every pair of homologous chromosomes is coming together, how many bivalents would be formed in a human cell? So how many homologous chromosome pairs could be formed? 23. We would have 23 pairings that have to come together. Interestingly, like the X chromosomes can find each other pretty easily, but even the X and the Y, they have a small region on them that allows them to find each other. So even though they don't have exactly homologous information, there's a region at the end that allows them to find each other. So they pair up in a process that's called synapsis. So this term right here goes into the blank, and then they physically swap chunks of DNA, which means there has to be a double strand break, and one of those strands invades the other strand. We don't go through the details of this class, but it's an intentional double strand break and then an invasion of the sister chromatid. In order to form this is homologous recombination that generates the crossover. The proteins that are involved in forming this are called the synaptonemal complex proteins. So in here, you see there are these, they look like a zipper. So that is actually the proteins that come together, bring the 2 chromosomes together, they zip up, kind of forming along the length. They recruit the enzymes that actually induce a double strand break, and then one of them will invade the other strand, and they're exchanging information. So this shouldn't leave any kind of damage or anything. It should resolve, but it's just swapping the two strands. And then they seal up and they fix the backbone. So at the end of the day, you should have no evidence that there was a double strand break, but you will then create like a hybrid strand of DNA. All right, so this first division then, I'm gonna go back to the picture here. Metaphase. So again, I want you to, with your neighbors, think about what's different between this metaphase and then the metaphase that we talked about with mitosis.
I don't know how the metaphase looks like for the other one.
I think they went 2B break.
Oh, yeah.
Because in mitosis, there's just it's 2 chromosomes thick. This one's 4 chromosomes.
Oh, that makes sense. Okay, I see. So I guess like the size difference.
Okay, so what's the distinguishing
There's an extra one.
difference? So you could recognize, if I showed you a picture of this, you could tell me it was meiosis 1. How do you know it's meiosis 1? Yeah, yeah.
The homologous pairs.
Yeah. What's lined up is the homologous pairs, right? So we have tetrads lined up. If we looked at mitosis, how would it look different? There would be like in single file, right? So we have double file. It would be single file. In the human chromosome, it would be 46 chromosomes lined up versus 23 pairs lined up. Okay, so that's the main distinguishing factor about metaphase 1. So then when we get to anaphase, what is separating is also different. When we talked about anaphase in mitosis, what actually split? The two sisters came apart. In this one, the sisters are staying together, but it's the homologous pairs that are coming apart. So we actually, I forgot I was going to do a little review about the metaphase to anaphase checkpoint, about how these come apart. But I might still do that review. What is it that is holding the two sisters together? Does anybody remember the name of the protein? Cohesin is that kind of belt-like protein that holds them together. So if we go back to that metaphase to anaphase checkpoint, they're held together until what event happens? Like, what are they waiting for before they can go through the checkpoint? Does anybody remember?
Oh, the spindles are attached.
Yeah, they have to be attached to have bipolar attachments, meaning that there's tension on both sides. That tells you that both of the sides of both kinetochores are attached to opposite poles. After that happens, remember there's a protein that is going to cut the cohesin. Anybody remember the name of that protein? Separase. And there's something that prevents it from activating. Securin. Yeah. So securin is blocking. It's like a scissor block. And then as soon as all of the attachments are there, anybody remember what happens where you kind of say, okay, now you can go, you can cut.
APC tags it, securin, ubiquitin, and then separate.
Yeah, so the ubiquitin is like that destruction tag. And so APC adds ubiquitin to securin that chews it up, and then separase can separate. So here, we're not gonna have that happen, right? We have to have a way to keep the sisters together. So let's look at what happens to keep the sisters together. Do you have a question?
Oh yeah, so that checkpoint, that only happens in mitosis.
So it does happen in both, but I'll show you there's a way to make sure that the sisters will stay together. But actually the checkpoint does happen in both. Again here, there's cohesin, which this is taken from a paper, so that's why they call it Rec 8, but you just have to know cohesin. Again, it's holding these all together. There's a protein, a special protein that is present in meiosis, and it's called shigoshin. I like this because the word Shigoshin is a Japanese word that means protective or guardian spirit or something like that. Yeah, guardian spirit. So Shigoshin, this is the guardian spirit of the sister chromatids. So what happens in order for cohesin to be cleaved, it actually has to get phosphorylated. That's a detail that we didn't have in the last lecture, but that has to be phosphorylated in order to be cleaved. What Shigoshin does is it recruits in phosphatases that take off the phosphate. And when it does that, it's protecting the sisters so that the sisters don't come apart. But these homologs can then be pulled apart, but it doesn't allow the two sisters to come apart, if that makes sense. So for this first blank, it should just be Shigoshin. Shigoshin prevents cohesin cleavage. On the next slide, I kind of show this phosphate piece, which is that phosphates get added to cohesin, and normally the phosphorylated cohesin is what can get cleaved, but Shigoshin recruits in a phosphatase. PP2A is a phosphatase, which we know phosphatases, they take off the phosphates. So when you do that, you prevent these two from separating. So for this blank, kinases phosphorylate cohesin, and phosphorylated cohesin is cleaved by separates, but Shigoshin protects the cohesin at the centromere by recruiting phosphatase. And that means that you're not going to separate those two strands. So the other thing to note is that part of what holds these two together, you can see they have that chiasmata crossover. That gets resolved right as these are pulling apart. So part of what holds them together, the homologs, is actually the crossover event. That's resolved to allow them to separate, to go through anaphase. All right, does anybody have any questions here? So now you can see like there's differences at every single stage. We talked about prophase, metaphase is what lines up. Anaphase is this whole thing that is preventing the sisters from coming apart. All right, so now we're done with meiosis 1. And now in those cells, they have chromosomes. Actually, let's go back and look at the numbers for this first one, so that we keep track of the numbers. So in the cell, how many chromosomes and how many chromatids do you see?
6 chromosomes, 12.
Everybody agree? 6 chromosomes. So the N number is what? 3 cents, right? Yeah. So there are 3 different sizes. They tried to show you different. I like to do that too. So you can use 2 different colors to represent maternal, paternal, and then here they have large, medium, and small. So there's 3 unique chromosomes, and in that picture there's still the chromatids. Now when you go through metaphase and anaphase, we get to meiosis 2. Now how many chromosomes are here? And how many chromatids?
6 chromatids.
Yeah, 3 chromosomes and 6 chromatids. This series of events is very similar to mitosis, right? Because you see there's no more crossing over. Crossing over does not happen in prophase 2. In metaphase, just individual chromosomes line up along the metaphase plate, and then those chromosomes, those sister chromatids, separate in anaphase, and then at the end we create haploid. So this starts out haploid, and it ends haploid, but the only thing that happens is the sisters separate. So if I gave you this picture, could you tell that it was meiosis 2 and not mitosis? How would you be able to tell me that that was a prophase cell from meiosis 2 and know that it's not from, yeah, can you?
Because of the crossover.
You see, yeah, so 1, you see evidence that crossing over happened. What else?
There are no homologous chromosomes.
Yeah, there's no homologous. If you count up the number of chromosomes, if I show you the picture, I will tell you what 2n is equal to. you could figure out that, oh, this one is haploid, so that must be in meiosis too. Okay. Yeah.
In telophase 1, does the nuclear envelope reform or does it just stay away?
No, that's a good question. I think in some species it does reform and others it doesn't. So there is a variety of whether, it makes sense to not waste the time to do that. But yeah, I have read that various species either reform or don't take the time to do that. Okay, so now we have enough information to do, you can do number 1 and number 2. So I feel like we can do these two here. For this first one, it says N is equal to 42. This is a huge number, right? So then you have to think about, it still is a diploid organism. So then you have to figure out what is diploid number. And for each of these, you're figuring out chromosomes and the total number of DNA molecules. That's another word for chromatid. So chromosome, chromatid at the beginning, chromosome, chromatid at the end. I'll let you work on it and kind of check each other, and then we can go over this one and make sure everybody's got it down.
Okay, so mitosis comes from 2 N to 2 N. Meiosis comes from 2 N to 2 N to 1. Meiosis 2 goes to N. So the total number of chromosomes at the beginning, we can say is, for mitosis, 84, because that gets replicated. And then meiosis 1 is 84 is 42.
What do you think for the total number of DNA molecules?
Is that asking for the, I don't know what that is. Is it the, is it like the end of how much there is? Or is it just? I was like, I don't know. Is it the chromosomes?
That's why I was wondering, like, what is the beginning stage? Is it like a interface or is it like?
Is it like, I don't know, what is it?
What's it asking for? The total number of DNA molecules?
Oh, yeah, so chromatid.
Yeah.
Oh, the
Yeah,
chromatids.
yeah.
Okay.
Yeah.
Alright.
I was like, is it the chromatid? I don't know how to describe it.
Okay.
It's like the thing.
Yeah, it's the number
It's the number of
individual.
individual.
Okay, yeah, so for this, if N is equal to 42, we're looking at a diploid. There are 180 in a diploid. Double, it would be double that. So 84. So you can
And
write
then
2 N
so
is equal
this
to
one,
84 at the top of the
it would
chart
be
here.
double of 84.
I think so.
Okay,
That's
and so
168.
I kind of started you off by saying that the ploidy at the beginning and the end is
Yeah,
going
that
to
should
be 84. So
Also
if we know that
168,
2N, they're going to be 2N. That tells us how many
Yeah.
chromosomes there should be.
Yeah.
84.
Yeah, because it doesn't really change that much from
Yeah.
mitosis.
This is
And then at the
doubles
end, they're
the
still
number, but then it
diploids.
has-
They
yeah,
still
I
have
know,
84.
so yeah,
Mhm.
okay.
But at the beginning, they have sisters. So each of these 84 has a duplicate. So we would double that again.
Yeah.
Figure out.
For, wait, mitosis, it goes from
So
N to N,
168, right?
right?
And then at the end, what
Or
happens
meiosis
is those two come apart.
Yeah.
End to end,
And
okay.
it's like-
Meiosis one goes from 2 on
So
10.
then we're back
Yeah,
to
two
Two
on
goes
ten,
from 10.
and N.
or 84. So
Wait,
those
no, that's not right.
168 split, so now we have 84 in each one. Yeah, so then meiosis one starts out the same. So it starts with 84. All of them are duplicated, so 168. And it's a
But
good
then,
way to
wait.
start. Okay, and then it's at the end, remember we said, I think I wrote it there, oh, I erased it.
Okay.
The reduction division happens, so you go from diploid to haploid. So this
And then
becomes
for
one
the
end.
total number of, the
if
chromatids
it's
for the
haploid,
meiosis
what is that?
one, it's going to be 84, same thing, or is it?
I think so.
Okay.
Yep, but
That's what I put.
what separated was these homologous chromosomes, so they still have sisters. So this, each of these still have
Because
sisters.
basically meiosis one,
Yeah.
it starts as a
Yeah.
cell.
And then you can try to figure out the rest, so this is
Diploid,
gonna
and
then
start,
into-
you know, the end of this is the beginning.
Yeah,
And
I have
then
And then
meiosis
see if
2
you
just
can figure out what
duplicates
would
the
happen
haploid.
at the very end.
Like,
Yeah, so it's gonna stay haploid, oh, it stays haploid, so haploid, haploid. Because the goal would be to make a sperm or an egg that has haploid at the very end.
I think,
And this is also
I feel
haploid.
like, I don't know, wait.
So
I think
these
meiosis
numbers,
2,
because that's the end
the
and that's
D
what
and
you
that should
start
be, that should be
with, these are
42.
the same.
Okay.
Because in
But then
meiosis
you
2,
just
you
have
have
to figure
the
out what
chromosomes
happens.
line up and then they
You know,
get
we know
split
haploid,
apart.
haploid,
Yeah.
this
Sister
number is
chromatid.
gonna
Yeah.
change,
Yeah, so that goes down by half again.
right? But what is gonna change?
Meaning that it has a 1
1, 1.
This number,
Yeah.
so what is, what happens with that?
That's it.
So will it double, or so you have these, and then at the end, they, oh, like you have a picture where you have these and they come apart. So they split, so you have sisters and then you have individual chromosomes, so it shouldn't get bigger. This number will divide by 2 to give you 42. Yeah, so this one is the beginning of it. They line up, they divide, and then at the end, we're looking at these, they have individual strands instead of sisters. Yeah, so that would be 42. Yeah. This one is just one chromosome. The chromosome, yeah, in this, we're not really looking at sets. That would be one chromosome, but it has 2 sister chromatids. Yes. Yeah. In this one, we're not actually looking at like a cell with multiple chromosomes. If you go to that picture that he's looking at, the meiosis too. Now we have,
Oh, it's
this
not.
is one set, because this is at meiosis 2 and that's
Never mind.
half.
Because
It's
I wrote,
one
I
set, so it
wrote.
has a big chromosome, a medium, and a small. So, yeah, so that would be,
I don't know.
yeah.
That makes more
So
sense.
very contrast. Oh, no, that makes
I
more
just wrote yes,
sense.
no.
I was like, should I write the face? Oh, you know what? Whatever.
So can you say again?
Probably that's gonna be better for me.
No, meiosis 1 still goes through cell division.
Hmm? Oh, it does?
Because it splits.
Yeah, this is chromatid.
Wait, what?
This is another.
It doesn't show it there, but like,
Chromatid.
it cleaves like normal, so you end up with 2
Oh,
cells.
she's, oh.
So it still goes through
So
cytokinesis.
it's,
So you're gonna
okay,
be going, you
gotcha.
go from
Yeah.
2 and
That's
actually start from
confusing.
the beginning of
Okay.
the next diploid. And then you go into the, anytime it changes, is going from this one to this one. And then it stays haploid, the whole rest of the cut, because the goal is to get it to be haploid. In the, I think I said that when you're looking at the life cycle with the eggs.
Hold on, I don't, how the heck do you know? Did you say that? How did I
But this
know?
is
Like, I
really,
feel like I missed so much things.
So it's 2 into 1 end, and then it stays 1 into 1 end.
Yes.
The very end.
I just assume.
Okay.
I don't know for sure. Like it has to, right?
I don't
Makes
know.
sense.
Oh, there's
I guess it
four
makes sense
cells.
now that you say it,
Oh,
but I don't
see.
know
There's four
why.
cells, but each cell has
How
one
those,
cell.
okay.
So it's still halfway.
So this is.
One, yeah, halfway, one end. Yeah.
Oh,
And then for the bottom, for the bottom table, some of them are just like yes or no, like if it says cell division, you say yes, it undergoes cell division. You could write the name of it, like that it's reduction or equational. If it says chromosome reduction, it's saying, does the ploidy change? That's what that means. So you say yes or no. And then a lot of those are, crossing over, you say yes or no. Yes, it happens, or no, it doesn't happen. Did you have a question?
I mean, even if it's not, like, two sister chromosomes, you still have
So like, which one are you looking at with this? Yes. Yeah, this is right.
Okay, so even if.
I think that's supposed to be 68.
oh,
Strand, yeah. It's still called a chromosome.
Right.
oh.
Like in G1,
I forgot about
we
that.
still count those
That
as
was-
chromosomes. They haven't been duplicated yet.
Yeah,
Oh, mine's up
Yeah.
in the
so there would be 84.
Wait,
Yeah.
what's the difference between the
Yeah,
crossing
and so
over
the
and
only
this?
one for this, because it's diploid, this should be
I think
84.
I
And
just
then
interpreted it
this
as
should be double
result,
84.
like for the
But yeah, the
random
pattern
distribution,
for
it
the
says,
rest
does
of
it
them is
result
good.
in random distribution?
Oh, okay.
So I think it's basically the same, except also yes for meiosis too.
This is
Like,
Figure it out.
I haven't known yes.
You
Because
did.
mitosis, it doesn't have.
Yeah, it doesn't have any, yeah.
Meiosis undergoes crossing over,
Yeah,
so it has to.
it has to.
In the meiosis two,
I mean, at the end, I
it
would.
previously went under, yeah,
Mhm,
so
but
it
then, like,
also,
Yeah.
So is this?
that's how I interpreted it. We'll find out how we might.
I mean, we can ask.
This is called a
I'll probably
chromosome.
ask after this.
Okay, that's 640.
I didn't quite know what to put for the last two, but I think I came up with
Yeah.
And
And
also-
appropriate answers.
Mitosis is just the, I don't know how to say it.
Yeah, and sometimes I like
Put
to
it up
draw
on
that picture, like, I just draw the sisters here, and then here I draw the tetrad, and then I can remember, like, okay, when they split, this still has sisters, and then here I'd have just a single pair of sisters.
But here,
They
like,
split.
oh, oh, okay, now I see, okay.
It's like,
I was like, oh, what do I have to say it? But then,
Are there like two
I don't know what I'm trying to say.
Yeah, so if we're looking like before cytokinesis happens, technically they're still in the same cell. That's kind of a time
Oh, I
in
know.
limbo where, what do you call it? Because that's one chromosome, but there's two copies of it, they're just not together.
Harry Potter.
So it's hard until we look at the end after cytokinesis has happened, because then we can clearly say there are 4 chromosomes and 4 chromatids in that cell. Or I don't know how to do that. Okay. Yeah.
I don't know.
Yeah, it does get jumbled in your head, I don't know.
So this one, the sister chromatid doesn't separate, right? So,
It
is
comes-
this
All I see
yeah,
are
that's the one where there are two.
Yeah, it comes
chromosomes,
with
and
it.
the chromosomes
Okay.
stay separate.
Gotcha.
Both the others, the chromosomes on evidence, they separate into chromatids.
Oh, so,
I think between like,
it would be
that
double
makes sense.
then.
And
So
like,
2 entities,
Well,
and then you would double that.
Oh, I can't forget it.